I request Dr. Riya Ratna to be on the desk.
Good afternoon. So we have Dr. Somal Sheth, retina surgeon, senior retina surgeon from Mumbai, and we have Professor Meenu from, she's a professor of ophthalmology and superintendent from Somaya Medical College and Hospital. और किरत मोदी सर क्या हो गया आ रहा है okay so and Takeshi will join in 1 or 2 minutes. In the meantime I request Dr. Riya Ratna on behalf of Dr. Umang Mathur to start the talk. Thank you. You want to say something? Yes.
Oh, so I think your talk is on unique population in India for vision research, right? So it should be unique and large. We have the numbers.
Yes.
Right?
Yes.
Please do carry.
And that's actually one of my first slides, and I'm starting with that point.
Yeah,
itself.
there it goes. Yes.
Yes. So hello everyone to the afternoon session, and I hope everyone's well fed and ready for it. I'm just going to dive right into it. So I would like to call them apparently rare diseases. So when we talk about genetic disorders and unique populations, we think about rare and unknown disorders of sorts. But anything that is rare in most of the surveys and by population studies abroad, just because of sheer numbers and just because of our population, which is now 1.4 trillion, becomes more common in our population, and we see it more often. The chart on the right side just shows numbers of different disorders that we at Shroff Eye Center see annually. And so 70 to 96 million people of these rare diseases are present in the country. And one very large example of it is MAC, the microphthalmos anophthalmos coloboma disorder. There were 545 patients noted in 1 year, which is the maximum in the world under the Bodhya Eye Consortium. And it was a massive study. It was published this year and last year, and Dr. Shailja Tibrewal was heading it. And that just shows the fact that these aren't so rare anymore. However, why should these be the center of vision research? So we have felt that there's a lack of current intervention in these. The average time to diagnose a rare or unique disease is 8 years in India. So people would stumble from one specialist to the other just to get a diagnosis. And there's a lack of disease leaders and collaborative workforces. So making cross referrals and such is not so common anymore. And there's a lack of deep phenotyping of disorders. So disorders such as retinitis pigmentosa or MAC, which have a broad spectrum of phenotype, patients need to undergo a lot of surveillance and a lot of evaluations to reach a specific diagnosis. And that actually helps us in the genetic diagnosis as well. And apart from all of the scientific reasons, there's also the personal toll that it takes, that individuals with these diseases, they remain without help and hope. So from here on, I am going to highlight the unique subsets of the known disorders in North India that we have noted, not everything, but just a few, keeping time in mind. So one is a subgroup of retinoblastoma. Going over what retinoblastoma is just quickly, it's a malignant yet curable form of retinal tumor in young children with mean survival age of 96% in developed countries and only 62% in developing countries like India, due to reasons like late diagnosis, inavailability of treatment, and just the inadequacy in the systems. So identification of RB1 germline status differentiates between sporadic and heritable RB. Now, RB is a curable form of retinoblastoma. It is also a form of a type of cancer which can be prevented from going from one generation to the next. Hence, looking at it and doing genetic testing becomes more relevant in these cases. And it will have an impact on prognostication and surveillance strategies, and which will result likely to increase the survival rate. So this is my own data of about 120 patients. And I'm going to highlight the genotype-phenotype correlation of a subset of RB patients. So the molecular profile that was observed, which is consistent with world literature, is that between a sample size of nearly equal bilateral and unilateral RB patients, 30% has hereditary disease. And among the bilateral individuals with healthy parents, only 3% were silent carriers, which basically means that they had the pathogenic mutation. However, the disease was not present in the parents. And the mean age of onset was much earlier in bilateral cases of 13 months, and unilateral cases was later, till an average of about 34 months. What was inconsistent, however, with the world literature in our population was that mutation detection rate was about 77% for bilateral patients, and the world literature says it's 90%. So where is the lag? And we will address that, and I'll address it with the kind of family that I have. So this is a family of multiple affected members. Started with an affected father, and all of their children are affected. They were diagnosed with bilateral group B RB at 1 year of age. That is about RIFA. And the father and sister were also known cases of retinoblastoma. Now, what are the investigations we performed for them? So RB1 gene analysis was done by NGS, which had 100% coverage of the gene. For RB1, what is known as being majorly for heritable cases, only need to screen retinoblastoma gene, which is RB1, but no variant was identified. Another type of change that could have happened in the gene was the deletion duplication changes that could be analyzed by MLPA. The same test was done. Again, no variant was identified. We then went on to test the other sibling just to see if there's a discrepancy. And this time we went for clinical exome sequencing, which basically encompasses more genes than just the RB1 gene, because we were interested to see, is there something else, any other oncogene, proto-oncogene, tumor suppressor gene that is causing this retinoblastoma. And they found a change in SDHD gene. I will talk about this gene in the next slide. So what we did was, we did not know whether this is related to what we are seeing in the disease. Is this the reason why the disease is happening in the family? So we went and checked all the affected and unaffected members, and the gene was co-segregating with the affection status, which means that the affected members of the family had this change in the SDHD gene, and the unaffected members, like the mother, it was not present. So we tried to look up world literature to see if there's any link with any changes seen in this gene with the pathogenesis of retinoblastoma, and no such link was identified. The role of SDHD gene is to cause pheocytochroma paraganglioma type 1. However, this phenotype was also not observed in our patient. So I took this family up because this is one of the many families now, around 5 bilateral hereditary families, 3 generations affected, and we cannot find mutations in RB1. So this is on average the difference we are finding. This is a unique population. There's a bilateral familial retinoblastoma, and there's bilateral sporadic retinoblastoma. So the average age of onset in our familial cases is much lower. It's less than 12 months, whereas in sporadic bilateral RB, it's variable and goes up to 5 years of age. There is a higher frequency of recurrence noted within the eye and also among family members, and a lower frequency in the sporadic cases. And the onset with advanced grade tumors is more in bilateral familial, and variable grade tumors are present in bilateral sporadic RB. However, when we did genetic testing and tried to match the mutation profile, what that means is the different kind of mutations that can happen in RB1 gene, like frame shift mutations, nonsense mutations, missense mutations, deletions, there was no correlation with the difference in the phenotype that we were observing. So we went to see if there are other people who have worked on such populations, if such an observation has been made by other people around the globe. So this was a study, it's a very new study published in 2021, which is titled A High-Risk Retinoblastoma Subset with Stemness Features, Dedifferentiated Cone State, and Neuronal Ganglional Cells Gene Expression. So what they basically did was they did omics analysis for 58 RB tumor samples to create subsets. And these studies included methylomics, proteomics, transcriptomics. I would like to pause and point that this means that now even in our understanding of retinoblastoma, we are trying to go beyond RB1 and germline mutations. There is a need, these tumors act differently, and there's a need to understand what's happening in real time in these tumors that is causing different phenotypes for our patients. So based on that, they made 2 subsets of patients. And the first subset created early onset heritable form, and the subset 2 consisted of non-heritable, less aggressive forms. Subset 1, they noted that tumors were expressed by mature cone markers. Is that for 10 minutes or 20 minutes? Okay. Okay. Okay, done. I'll just wrap it up in 2. Okay. And all right. And in subset 2, they were markers of cells of neuronal and ganglion cell origin. So the cell of origin from where these tumors are beginning are different for these patients. There was interaction of EF2, MYCN genes found in subset 2, which was not present in subset 1. And there was a higher propensity of metastasis, which is phenotypically consistent with what is known in the unilateral non-heritable cases than in the lower propensity of metastasis was known in the other subset. The reason I brought out this study is because this gives us direction to now target our unique population. The question marks that we are getting in our population of retinoblastoma patients, these are probably factors that are differentiating them from the other ones. So current need, what is the current need then? According to the TNM system by AJCC, they now include hereditary in their classification, and which says that we should be screening all patients of retinoblastoma, irrespective of whether they are unilateral, bilateral, with family history, without. And there needs to be a tumor mutation profile registry. So any person who's performing what activities are happening within the tumor should be pointing out. And that this has proven that we need to look beyond RB1 to target these patients. So I think I will skip through this, but I will just speak about it in a minute. There's a subset of population of microphthalmos and ophthalmos coloboma. It's a polygenic disorder. A lot of genes are noted. The subset of population that we noted recently is we made surveillance queries and found out that patients with maternal vitamin A deficiency, mothers who had vitamin A deficiency had children with microphthalmos disorders, and that frequency was higher. So we postulate that there could be a pathway in the retinoic acid. There could be genes in the retinoic acid pathway that could lead to this disorder, and those need to be looked at. So with that, I think I will hand it over to Dr.
Yeah, we welcome Dr. Kirit Modi sir on the dais. The second leg of the talk will be presented by none other than Professor Natarajan.
And thank you, Dr. Ria.
I think is Takeshi come back? Can I just check the overall online? Takeshi, I hope you had a lunch and rest of your Namrata there. Has Namrata logged in? Can you check?
Dr. Takeshi is there already, I think.
Okay.
I can see him.
And Namrata? The next speaker is Namrata, and then Kelvin Pang is there. But yeah, hi, Kelvin. Hi. So I think we are in time, and I hope, Kelvin, you had lunch. John is waiting here, and he's there whole day. Takeshi and Kelvin, and I said, John, he has to prepare some slides. He will be working in the background, and I'm here. So I'll give my talk, Unique Population in India, continuation along with Dr. Umang Mathur and Ria for vision research, genetic retinal diseases. And as per WHO, genetic eye disorder are one of the top 10 major causes of global ocular health burden. And GRD was defined as the disease of the retina having possible or definite genetic etiology. and a systemic assessment of ophthalmology and vision-related research publication from India are very limited, and particularly the analysis of the research on genetic retinal diseases is not available. And this is one of the publication which we have worked together in the Advances in Vision Research Volume 2. And without objective information about current research output, it is difficult to plan for necessary improvements in the infrastructure related to the understanding, treatment, and prevention of eye diseases. ARMD, diabetic retinopathy, take the major share of the adult eye diseases component that particularly affects the neurovascular retina. And AMD, all of us know you have the dry AMD with drusen and the wet AMD with the submacular hemorrhage. And both are adult onset retinal diseases, and the prevalence of AMD is 0.05% under age 50 years and 12% above 80 years, with a rising estimation of 196 million in 2020 to 288 million by 2040, and with a higher prevalence among Europeans compared to the Asians. In India, diabetic retinopathy prevalence is around 15 to 18% among the diabetic population over 40 years of age. And genetic factors play an important role in the pathogenesis of diabetic retinopathy, the burden of which is on a rise in the working age population. And the rates of different inherited retinal diseases vary. Monogenic vitreoretinal diseases have a prevalence in 1 in 2,000 individual. The prevalence of RP is between 1 in 3,000 to 1 in 5,000 individual, and choroideremia has an incidence rate of 1 in 50,000. And there is a gene therapy for RP265 mutation and choroideremia in a big way, which is happening in the Europe and US. And more than 100 genes are known to cause Mendelian types of retinal degeneration, including syndromic and non-syndromic RP. It is presumed that this constitutes only 60% of all the genes known so far, and remaining are yet to be identified. And that's why we are all assembled world over in this first genetic conference in GEGC. I hope we'll have the next GEGC meeting, international meeting, post RO will be, I hope, will be done by John Prakash, and definitely myself and Takeshi will work for it. Many significant genes, including RP65, have been identified from this region and are associated with consanguineous autosomal recessive pedigrees. I spoke to the Spark Therapeutics in Hamburg, and they told me they cannot come to India because of they have an agreement with Novartis, and Novartis also said they can't talk about RP65 mutation and the Spark Therapeutics and the Luxturna because they are not approved by Government of India. But it's available in the US, and I'm giving a hint where the Global Eye Genetics Consortium will approach the Basel. And yesterday Raghu Modi suggested, and we should ask them to come and do the second GEGC meeting in Washington. Mendelian diseases such as RP, congenital stationary night blindness, and Stargardt disease have been reported to caused by pathogenic gene mutation. And prevalence of RP is 1 in 930 urban South India and 1 in 372 in rural India. The reason for such a prevalence of RP is not clear, but could be due to consanguineous marital practice, which happened in the past widely, but it will continue till it is diluted by various different category of marriages. And interestingly, 4 of the top 6 institutes are located in southern part of India: the Shankar Nethralaya, LV Prasad, Arvind Eye Hospital, Narayan Nethralaya. The rest to the PGI Chandigarh and RP Center in northern part of India. And I'm happy Western India we have adityajyot. And these institutes, sharing their resources and infrastructure, may undertake projects in ocular genetics, mentoring and collaborating with both existing and developing eye research institute from other parts of the country to bridge the gap in the field of research in ophthalmology, particularly in genetics. And RB, we had a lot of talks yesterday in the Women in Ophthalmology, and the genetic lab in India started in 2000 and are on surge due to the generous support from the different government agencies, the DBT, the department, DST, and ICMR. And however, the labs conducting dedicated research in the field of the GRD are limited in India. There is an urgent need for increasing the number. Collaboration between the DBT, India, and the National Institute, US, is an important step in the progress of research, even though both the President of the United States and the Prime Minister of India have done that last few years. And thanks to Dr. John Prakash, who has expanded the horizon of doing the Indo-American collaboration. And there are a lot of Indo-European collaborations available, but I think we have to use that. And we are doing this community DR screening. So earlier it was Adityajyot Foundation for Twinkling Little Eyes. Now it is Kamala Sundaram Foundation in my parents' name. And we have come in the Guinness record. And we also have the first eye screening on wheels in the world, which is inaugurated by my late grandmother, whose photographs I've kept here. And that's why I use the word ABCD, and same anybody can screen for diabetic retinopathy and use this stop blindness program. The reason I'm talking about this is we have published this in JAMA, but we can actually, we are also working with Case Western University. Thanks to Dr. Sudha Iyengar, everybody in genetics knows her, and we are trying to find a unique population in Dharavi. And we have the project going on, and I hope whether DBTQs or Indo-American funds I get, I have decided, and I'm starting the project on 1st December. So we can make India free of the burden of diabetic blindness, and teleophthalmology can show us the way. And I also decided I'm going to apply, Adityajyot will apply for a BIRAC, Government of India, and hope Bombay Hospital, the Somaya Medical College, and whoever wants to join, will start the clinical registry for inherited retinal degeneration. Thanks to Dr. Jaydeep, Afroz, and Samir for this presentation. And can I have Dr. Takeshi? Is he back? I know he's back. He can give his comments. And Dr. Kirit Modi is here. He's the youngest ophthalmologist here. And plus, I want to tell him that he's supporting me from the day I came to Bombay, and that is today, November 20th, 1988, I was here. My son is also born today. Today is his birthday. Thank you, Dr. Kirit Modi, sir, for blessing me on the first day and also today, sir. Dr. Modi, sir. And yesterday we had Raghu Modi, and today we have Kirit Modi. And Takeshi, you can have some comments.
Do you know why is this RP65 trial is not approved in India? What's the reason?
No, I think it is a paperwork problem, because the government, the company has to submit their details to the DCGI, that is the Department of for the Drug Control of India, and then it has to come.
The reason I'm asking is that it's already approved by 40 countries already.
Yeah.
And
why
the most populated?
I forget her name. I have it in my mobile. I met her in Hamburg during the URETNA meeting, and I told the importance of GEGC meeting. The idea, that's my original idea of joining the AEGC and GEGC, is to bring gene therapy to India, and whole world should use it. And I think we have almost a few million patients of a genetic eye disease in India, and we have to do something. And I'm still keeping your idea of replicating the National Institute of Sense Organs in Mumbai, and Dr. Takeshi will be the director. And I'm only 65 young, and working with 18-year energy. And I need a blessing of Dr. Kirit Modi for that. And Dr. Kirit Modi can also sponsor the research. Sir, you can give us some money. Yes, sir. You want to say something?
I just wanted to ask you a question, Dr. Natarajan.
Yes, sir.
How we are a unique population in India for genetic studies? Can you tell me?
Yes, sir, that's what we have. Actually, the AIMS had done a study in Rajasthan. One group of whatever a sect of family do not get diabetes at all. So like that, we don't know still why. Similarly, only 25% of diabetic patients will get diabetic retinopathy, and we don't know why still. And Shankaranetla has done a publication, diabetic retinopathy with more than 15 years of duration, and diabetic retinopathy with less than 5 years of diabetes. So still we don't know the cause. What is that one which is protecting somebody from diabetic retinopathy and somebody? So even Dr. Skippen's Research Institute is finding out why everybody is not getting diabetic retinopathy, even though it's a microangiopathy. Somebody will get vascular retinopathy for in the vascular lesions in the brain, heart, kidney, or feet, neuropathy. But we don't know why, who gets what. Even the biggest research is done by Diabetic Institute of Dr. Mohan Vishwanathan, and they have got Modi gene. That's called Modi gene. I don't know what it is. but they are also still not knowing who will get diabetic retinopathy and who will not. So we are working on the unique population of diabetes in Dharavi. The reason is people are living for generations from all parts of India in Dharavi. So what is called as epigenetics has changed. Their diet, their environment, their way of working has changed. So we are hope to find something, but it will take another 8 years. That's what Dr. Sudhangar said. We are trying to find a biomarker, and Shankar, we did a Ornate India study where 20 institutes are involved, where we have found out that protein, if you do that, and I think those group of patients only may are prone for diabetic retinopathy. I don't remember the exact one. And that costs only 200 rupees, I think. But now in all India, we are contemplating whether to do that. So we have to find some way so that we don't have to screen all the 77 million diabetics we have. But we don't know what to do because we don't have that many doctors. That's why I'm using the mobile phone to screen. I mentioned only 2 slides about screening. So we have a unique population of having the maximum number of diabetes in the world, maximum number of retinal degenerations in the world, and maximum number of any genetic eye disease, retinal disease, JRD. Yes, sir. John. Sir, I know you can give us some money, sir. Kirit Modi, sir, you can give us some money for doing research.
Our next talk is
No, sir.
unique
Dr. John-
population in
Dr.
China.
John wants to say
Sir,
something.
it's speaking.
Yes, thank you so much. And this is really I want to point out 3 things. We've discussed a lot of things since yesterday all day, and then this morning many in 2 sessions we discussed several things. But I want to say 2 important things. Number 1 is it's not only the National Eye Institute in Washington or in America that does a fair amount of work here in this country, but also a large number of pretty much all institutes. We have 27 institutes and centers in within the NIH, and pretty much everyone is working here because of several reasons. Number 1, is that it's a unique population, large population. That's the number 1. Number 2 reason is that Indian scientists are just amazingly and most active and most energetic. And number 3 is just that there's just so many things happening in genetic research, and people are hungry for research, hungry for knowledge. And I feel very strongly that a lot of answers are going to come from India, not only answer to diabetic retinopathy and and other diseases. And the third thing for National Eye Institute is very, very important for me on a personal level to say that India. Third reason, and the most important reason, India has got Professor Natarajan, who gets things done. So obviously you can see this conference first, it was not an easy task. We were going to do it in Washington, DC. The whole world shut down in May of 2020. But Dr. Natarajan took it upon him, and he said that both of us decided that it is time to just get up and start moving. So it is not that how many thousand, how many dozen, or how many hundred, or how many thousand people are attending this, but it was very important for both of us, and Dr. Iwara and everybody else, and Dr. Calvin Pang, Paul Baird. It's very important. We decided that we are up and we are running, and Dr. Natarajan made it happen. So once again, a grand kudos to Dr. Natarajan for getting it done, doing it. And Dr. Iwata said, if anybody, he doesn't know how to fail. So I remember that Dr. Iwata said to me, Dr. Natarajan doesn't know how to fail. So he's exceeded all expectations, and he has tired me out. Honestly, I tell you, since I came to India, I've hardly slept, like only a few hours a day. So, but he's telling me, go and get some rest. And tomorrow morning, I'm flying to Aravind Eye Care System to Madurai. So he said, go and prepare your talk. So I'm going to hand this over to Dr. Takeshi Iwata and Dr. Natarajan to do the rest of the session. But I was waiting for this talk to happen. And officially, Dr. Takeshi, may I have your permission to go and prepare my talk and get some and do things? I have several talks I have to give. But Dr. Natarajan, do I have your official permission to do that? All right. Yeah. Very well done. Congratulations to the entire Dr. Natarajan team. Thank you so much.
Thank you.
Dr. Kirit Modi,
Thank
over
you,
to you,
John.
sir. Next is Kelvin Pang.
So we now have Dr. Kelvin Pang. who's going to be presenting a very similar topic to what we just did previously, unique population in China for vision research. Over to you, Dr. Kelvin Peng. We introduced him in the morning. Can we unmute him, please? He's not unmuted, I guess. He can only do it.
Alvin, we cannot hear you. Your mic is off.
Dr. Pang, can you unmute yourself, please? We cannot hear you. We can see your presentation, but we can hear you now.
Hello.
Yes, please go on. We can hear you.
Thanks very much. Sorry, there's some technical problem. Thank you very much. Wonderful to be here again. Actually, I stay with you almost all day. Not all the talks, most of the talks after in the morning. Yes, you're right. This is a similar topic as the previous one, but the way I present it is quite different. It's not going to details. I want to share some features and views about genetic eye studies in China in a wide perspective. Now, early in the morning, when I was introduced, so my friends in India know that I have a fairly wide collection with Indian friends of ophthalmologists in southern India and also in New Delhi. I visited before the pandemic. I visit India almost once every 2 years in all your major centers. So I can say that I myself know something about ophthalmology in India, right, and have personal collections with your top clinical clinician scientists. And I always invite Indian ophthalmologists on online discussions and presentation and lectures in Hong Kong. So that's the point I want to make, is that this morning, as well as my other experiences with Indian ophthalmologists and clinician scientists, that I'm always overwhelmed by what John Prakash had mentioned earlier on, that the amount of research and the depth of research in India, right, is tremendous. And because of that, I really want my Indian friends to have a bigger influence. I mean, you are already having a big influence, but then to be even more penetrative, influence in ophthalmology worldwide, right? For example, say in my center in Hong Kong, I always have international fellows from different parts of the world. So I would welcome more of your young Indian clinician scientists. Right? And I have strong connection with Singapore. I know, Singapore and India, and with specific eye centers in India have very strong connections. But I want it more, right? So Hong Kong, Singapore, and other parts of Southeast Asia, I think ophthalmology on the world stage, and we can say that Singapore and Hong Kong had maybe a bit more influence than the other countries or other regions in Southeast Asia. And on that basis, I want support and help, right, and supply of brilliant Indian visual scientists and ophthalmologists, like what I have encountered online this morning and my other experiences in India. So on that basis, I want to welcome our Indian friends to travel more, right, and also to have more collections with colleagues, in particularly in Southeast Asia. And on that, I also want to let you know a bit more about China in the sort of information that's related to collaborations. So unique population in China, I could easily say unique populations with an S here in China. Now this is where my patient base, where I work, right? Firstly, in Hong Kong. So in Hong Kong, we have population only about 7 million people. So I don't have many, starters. I only have 2 patients of LCA, Labor's and Morris's Labor's optic pneumorosis, right? Congenital amaurosis. Right now for many years. So Hong Kong is here, at the southern tip of China. We were a British colony, and of course in 1997 we returned to China, so it's still a special administration region in China. And because of that, you see this red line there, a bit thin, that there are restrictions of people coming into Hong Kong and from Hong Kong into China. Therefore, we still have a fairly stable population in Hong Kong. That means that, no matter how many hospitals that, my team are working on, for the whole of Hong Kong, we still have only about 7 million people. And from that, we start up our eye genetics work. I'll come back to that later. However, we have an eye center inside mainland China, the John Shantou International Eye Center. And so that means that, my team in Hong Kong have another team in Shantou, and we have access to patients from all over China. Right? Now that makes a lot of difference. Now China, of course, is the population is similar to India, right? So we could have patients from all over the country in Shantou, but it's not just that, or it's not that. Well, it doesn't really work that way, right? I'll come back to that again. Now, but then in Shantou, both Hong Kong and Shantou, we can say it is in southern China. So, but then in Shantou, there's also fairly stable population. Right, people with, that's in China, right? In this region, we call Chaoshan area around Shantou, which is only about 400 miles from Hong Kong, or 500 kilometers from Hong Kong, right? That in this region, about 20 million people having similar sort of lifestyle, right? And diet and habit, and they're fairly stable in this region. Therefore, I could have the Shantou cohorts as a validation cohort for my genetic studies. On the other hand, I could combine them so that together we form a South China cohort. So that's the convenience of working in China. Then I want my Indian friends to know that. So these are sort of geographic advantages, right? In Shantou, we have a stable population, and that happened in other regions of China. So I'll come back to that later. Right. So this is my patient base to let you know that in Hong Kong, we have fairly stable population. In terms of rare eye diseases, we are in great disadvantage, right? But inside mainland China, of course, rare eye diseases, you could have a lot more occurrences. And then in Shantou, we are completing a new hospital complex and going to be operational next year. So once again, I extend my invitation to all my friends in India to come and help us, and then to share the clinical materials and to collaborate. Right? So it's a fairly big hospital complex. Now, based in Hong Kong, right? That's the purple spot there. So I have collections with different parts of China, and so you certainly notice over the last, and particularly 10 years, there have been very high increasing number of genetic reports, eye diseases, right, from various parts of China, particularly from the major cities and major centers. So I have collection with all of them that, if you're interested, we can make collections. Meanwhile, in China, we have also built up, clinics in the sort of remote areas, right? You heard about this Tibet and then Xinjiang, these sort of remote areas, northwest and northeast. So we also have those. So from these eye centers, eye units, right, that we build, we can collect, patient sample. So based on Hong Kong, I have a strong collection and working with colleagues in inside mainland China, and of course with Singapore. And I have, friends collection with in India. And every eye center have collection with Morpheus, right? I used to do a lot of work with Decode in Iceland, right, in the early days of eye genomics. And so that was 15, 20 years ago. We published a few Nature Genetics papers together. And in America, so we have some various centers that we work closely together. And of course, with Paul Bird in Melbourne and Takashi in India, in Japan and Tokyo. And also I'm very close with Nacida, Koji Nacida and Motokazu in Osaka. And the blue spots are places that I have networked, but they're not close collaborations. So again, I want to let my Indian friends know that even based in Hong Kong, a small island, a small, population of 7 million, I collab with other places over the world, and that's essentially needed for eye genetics. And also, there are advantages in being in a small place, in a small city like Hong Kong with small population, in that transfer convenient and sampling is convenient. So for 20 years, we work on a whole spectrum of eye diseases, right? AMD, PCV, CSCL, and all forms of glaucoma, and of course, common eye diseases like myopia and refractive errors. And special interest in eye diseases, uveitis, and keratoconus. So on that, it's convenient that we set up similar protocols and technical support that we could work on whole spectrum of common eye diseases. But then for monogenic eye diseases, because we are of the small population, 7 million, we could only work on patients that we can get off. So these are the diseases that we work on. But then we also have started about 5-6 years now, quantitative ocular trace. And so again, we make good use of a small city, concrete and condensed, right? So we can collect data. So we could invite parents and children to come into our eye center in weekends and for 2 or 3 hours, and we do all these quantitative investigations, right? Keeping a family with us. So we have been doing this for about 8 years now. And for that, we have all this data from them, and also we follow them up. Again, there's an advantage of being in a city, in a small city, right, like Hong Kong, that communications and transport are convenient. And on that, we also do molecular genomics on this. So I want to let you know that in China, there are places like Hong Kong that we could do all this within a city, led by a major eye center with our eye hospitals all around in the city. Right? This is to let you know. Now come to the technologies. Now technologies have developed quite, of course, very, very advancing nowadays and very rigorously. And there also have been a very big change in the technology and in the strategy. For example, 20 years ago, when I started eye genetics, right? I had to have my own, sequencer, automated sequencer, and all these fundamental or basic or essential technologies. I got to have them, right? And then as time goes, I'm sure you are all aware of that nowadays we don't really need to install the high power technologies in our own laboratories because we source out. We pay for service providers, right? So right now in my lab, we still, of course, have to do sequencing. We will still do some next generation, sequencing. But GWAS, whole genome exome sequencing, whole genome sequencing, all this we send out to outside service providers in Korea, in America, in other parts, other regions where they are good, service supply. So this is a great advantage for us, to collaborate, right? That we don't need a high capital cost. We don't need to have our own high skilled technicians or scientists to work on these technologies, right? We just focus on our phenotyping, sampling, and interpretation results, statistics, and all this that have to be done by us. So I can see this is a great help to us. And because that, we can do multi-center studies and global multi-center studies globally as well. And then, of course, we need to our own visual scientists or collaboration with the molecular biologists to work on gene pathway molecular genetic biology studies. So nowadays, essentially, we don't need to install or to, run high technologies for molecular genomics, and that's a great advantage. I take an example of why we need collaboration. One example of myopia. So we know myopia is essentially the elongation of the eyeball, essentially. And then we now know that risk factors, mechanisms, interacting factors are extremely complicated, even the pathophysiology and along the natural history, because as myopia goes, well, it may be stationary or stable after about 30 years ago, but then the complications will come in, and can the complications due to high myopia in particular can linger on, can happen later in life. And of course, now we also have data to show that it's not just high myopia that has, serious complications. Even mild myopia also has increased in 1 or 2-fold or 2 or 3-fold, chance of getting glaucoma and other serious diseases later in life. So that's also very difficult to understand. How come this would linger on for so long and still have complications? So myopia is extremely complicated. But what we know about myopia is that it is both nature, and still a lot of question mark, because right now we still do not have one causative gene, right? Clear causative mechanism leading to even high myopia, except this syndromic high myopia. And then nurture sort of extrinsic factors are extremely difficult to quantitate. And now we are working on that because light sensors and we can quantitate outdoor activities. But then with myopia, we come to know a very important concept of inheritance, that parental effect on myopia in the children or even the grandchildren, right, are more than genetic, because it's also passing on transmission of lifestyle, and that plays an important role of the development of myopia, to serious myopia like high myopia. Right? And the lifestyle that's not just work by itself, it's also interactive. So again, right now, although we're working very vigorously over the last few years globally in many centers, but still we need more data to confirm, the so-called polygenic risk factor and then the actual risk interactive factors between, say, linear work and specific genes. So we still have a lot of work to do on that. Right? Therefore, in Hong Kong, based in Hong Kong and my eye center inside mainland China, we conduct, whole genome sequencing, with environment. So for myopia genetics is a whole lot of environmental work, lifestyle work, questionnaires, and also, a lot of these outside genetics that we have to work on. And actually, I haven't even mentioned epigenetics. So because of that, we really need data, and we need collaboration. Collaboration can be collaboration with a project and collaboration of protocols. I'm very happy to share with my collaborators, friends, protocols that we have validated in my eye centers in Hong Kong that my friends, collaborators can use and of course modify accordingly. But of course, if you have the fundamental, it's a lot quicker to go further, right? I want to come. So that's what is happening in myopia, that we do need global collaborations. I want to come back to China. Now, China, big country and a very populated country. China and India are the two most populated countries in the world. So in a sense that it's difficult to understand how this sort of occurrences, geographic effects on say genetic eye diseases. But I can tell you it may not be so complicated. Essentially, 95% of Chinese, like myself, claim ourselves as Hans, right? In terms of ethnic groups, so it's a major ethnic group in China, very dominated by Hans, right? But you can certainly, you can easily see that over the thousands of history of China, right? Inside China, right? You can the predominant ethnic group of Hans. Hans had to be very mixed. Right, so Hans itself is mixed. Now, essentially, based on HLA typing, we can differentiate Northern Hans and Southern Hans, essentially, right? And there are differences, and in occurrences and characteristics of many other genetic diseases, for example, inborn errors of metabolism, we do see not just phenotype, but also phenotypic differences between, say, patients in Northern China, like in Beijing, and patients like in my center in Hong Kong, right? So there are differences like that. So this is for the Hans. But in the meantime, apart from dominated by Hans in China, there are about 30, a bit more than 30 ethnic minorities, right? So that's what's happening, right? So in China, we can work with Hans, and the Hans can be in different places. But then, even for Hans or the minorities, we also have pockets of population. And so I mentioned in Shantou, we could have about 20 million of, say, Han Chinese in Shantou area, that they've been there for thousands of years. So that happened in the northwest of China, northeast of China, and different regions of China, right? So there are regional ethnic differentiation. More than that, say in Hong Kong, right? We essentially work on Hans. But then in Hong Kong, we have a very interesting ethnic minority. That's the Gurkhas. Now, that's not Chinese, right? There are Gurkhas, that's Nepalese population in Hong Kong. They're real Nepalese, right? But they grew up in Hong Kong. Therefore, in a place like China, you do have people born in a region and raised up in a region, but of different ethnic group. Therefore, I want to mention particularly the city of Shenzhen, that's adjacent to Hong Kong. Many of you have heard about this city of Shenzhen. It's the newest city in China, right? It's sort of city of cyber city. All the engineers, software engineers now, and high technologies are condensing themselves in Shenzhen. About billion population of 20 million people. Now, this Shenzhen population represent the whole of China. They were grown up in China, so grown up in Shenzhen, right? But then they are represent all over from over China.
Pang?
Yes.
Yeah. Could you please summarize, sir?
Yes, that's the. I come to the last two slides, right? Now, let me go to the next slide. Right. Now, I want to come to the two last slides, because that's what's happening in China. And that's why for GEGC, Global Eye Genetics Consortium, we have four of us: John, of course, our leader based in Washington, D.C., and then Takashi in Tokyo, I in Hong Kong, and Professor Natarajan in India, and Paul Bird in Melbourne. We could be anchors, right, for our study groups, our cohorts in this region. And in this region, we have a great advantage of having similar time difference, very little time difference. So I want to encourage all my friends, right, in South Asia, in Southeast Asia, and work together. And then we do need, a lot, as why I keep on mentioning data, that we need data. And in Europe, well, the CREAM consortium, and in America, one we mentioned by Professor Jenny Ricks, the labor consortium. So they're essentially Caucasians, right? But then we really need more data from Asia, particularly South Asia and Southeast Asia. So this is a tremendous conference that I want to encourage, all our friends in this region on eye genetics to work together. Thank you very much.
Thank you, Dr. Pang. Any question? Okay, so we would like to go on to the next talk.
Can I say one thing?
Yes.
Yes. So going and doing the analysis for the Japanese populations, we went, of course, with a target sequence. Eventually, we went to the whole exome sequence, and as I told you about our result, only 40% of the pedigree we had found with the whole exome. Then that's why we're now moving to the whole genome, but probably we can go probably up to like 50% or so. So the remaining pedigree will be completely blank. And those are the families I think we really need to collaborate with. So if you come up with the pedigree after your whole genome is done and still you cannot figure out, and I'm sure there's these kind of pedigrees there in Australia, in India, and elsewhere, we can get together why we cannot detect with the whole genome sequence. So I think those kind of project is probably a GGC type or maybe an EI fundable project. That's what I think.
Definitely, I'm step behind you, Takashi. Yes, these pedigrees we were working on whole genome sequencing. Yeah, we'll be working together, Takashi. Definitely, we need to.
Yeah, thank you.
Yeah, thank you, Dr. Takashi. That was a good point made. We now go on to the next talk by Dr. Namrata Sharma. We all know her, and we rather knew her as the Secretary General of All India Ophthalmic Society. And she's also the Regional Secretary at the Asia Pacific Academy of Ophthalmology. She's done her post-graduation from RP Center, and she's currently working in AIMS as well. So she has presented a lot on cataract and refractive surgeries worldwide. We also know her as a keen researcher and academician, and she will be presenting on a very baffling and an interesting topic on genetics of keratoconus. Dr. Namrata Sharma, are you there on Zoom? Okay. Dr. Namrata, we can't see you nor we can hear you. If you can make yourself visible and audible. Can you call her? Does she have a recorded talk? No, right? Does she have a recorded talk or she's going to go live, right? So any questions from the audience while we have Dr. Namrata go live? She probably will take a minute or so. Anyone from the Zoom meet also, if they have a question, they can raise it. I think she will just join shortly. We'll wait for a minute. If not, then we'll move on to the next speaker. Okay. What I'm suggesting is club it together. We present it and then we take this after. Yeah, we would request Dr. Tanya, the next speaker, to be ready with her talk. Dr. Natarajan has two talks, and I think we would merge both of them together so that he has one singular talk on both the talks combined together. So we would want Dr. Tanya, if you can contact her to be ready. Converse. Okay, fine. Is Dr. Namrata there now? Dr. Namrata, can you hear us? You have to unmute yourself. Yes, you can hear us, Dr. Namrata?
Yeah, I can hear you.
Yeah, so we've just introduced you actually. You can go on with your talk on keratoconus genetics.
Okay, thank you so much.
Welcome.
So, can you see my screen?
Yes,
Hello?
madam. You are visible and audible. Please carry on.
Thank you. So I would like to thank Dr. Natarajan for organizing this first international GEGC meeting. And I would be talking to you about genetics of corneal ectasias. So corneal ectasias can be both primary and can be even iatrogenic, and somewhere down the line, even the ones which are iatrogenic possibly have some genetic association. Primary can be keratoconus, keratoglobus, pellucid marginal degeneration, and Terrien's marginal degeneration. Now, keratoconus is bilateral, asymmetric, has irregular astigmatism, and it can be unilateral. And one study has shown that 41% of keratoconus patients were unilateral at the time of diagnosis. And the prevalence traditionally is reported from 50 to 230 per 1 lakh patients. Now, again, the ABCD classification helps to classify the keratoconus into various stages depending upon the severity. And why in some cases it is less severe and why it is more severe in some of the cases is again something which genetics can explain. In normal anatomy of cornea, I think everybody knows, but the components of the epithelial basement membrane are collagen, laminins, heparin sulfate, and etc. And the biochemical composition consists of the collagen and the collagen fibrils. And in keratoconic cornea, there's reduction of the epithelial and the stromal keratocytes, reduction of the epithelial and the stromal collagen content, and degradation of the corneal membranes and extracellular matrix, as well as abnormal mitochondrial functions which cause cell death. Coupled with that, there is deranged lipid metabolism, which is associated with stromal degeneration and disrupted epithelial integrity. At the molecular level, if you see, there's upregulation of the degradative enzymes such as acid esterase, acid phosphatase, cathepsins B and G, and some matrix metalloproteinases. And there's downregulation of the proteinase inhibitors. And there's 4 times increase in the receptors of interleukin-1, causing apoptosis of the stromal keratocytes. Coupled with that, there's microtrauma, which releases the interleukin-1 from the epithelium. Now, again, if we see the etiology of keratoconus is very complex, and although genetics is involved in it, but eye rubbing has a role to play because that possibly releases the inflammatory mediators, and there's upregulation of these inflammatory mediators. And sometimes there may not be the progression of the entire involvement of the entire cornea, but only focal progression may be there. And this may cause corneal thinning as well as steepening of the cornea. Now, there is a proposed pathway for keratoconus, which, like I said earlier, has not only the mechanical theory, but genetics coupled with it. And I think both of them have to be taken into consideration when we look at the pathogenesis of keratoconus. Now, there are specific genes which are associated with corneal biomechanics, central corneal thickness, corneal curvature, corneal dystrophies, keratoconus genes as such, and all these factors, they are interlinked with one another. And sometimes keratoconus is also associated with various other ocular syndromes, and there also there is sharing of the genes. So keratoconus genes specifically with no other ocular disease are which are shown here. And again, all these things have to be taken into consideration when looking at the genetics. The techniques for mapping of keratoconus genes include the linkage analysis, the genome-wide association studies, the next generation sequencing, as well as the copy and number variation, as well as mutational analysis. And this is our publication on mitochondrial complex 1 gene analysis in keratoconus, where we looked at 20 keratoconic corneas and 20 healthy controls. And we did find that there were 18 nucleotide variations and 2 novel frame shift mutations, which were detected in cases of keratoconus. And there is a mechanism by which there is increased reactive oxygen species, as well as oxidative stress signaling, which causes keratoconus. Now, if you look at the DNA sequence chromatogram, then you can see that there is deletion of nucleotide G codon, which has changed from GGC to GCT here. And there is deletion of nucleotide CA codon change, which has changed from ATC to ATT. We've also looked at the VSX1 gene analysis in cases of 50 cases of keratoconus, and the entire coding region of the VSX1 gene was amplified by the PCR, and sequencing analysis was done, which again showed 2 alterations which have been shown here, but there was no pathogenic VSX1 mutation which was identified. Now, again, the DNA sequence chromatogram will show the deletions of the specific codons and the changes thereof. And there are other genetic loci also, like 13q32, which may be involved in the pathogenesis of keratoconus. Now, genetic associations for keratoconus will help to provide insight into disease mechanism. It will help to identify the biomarkers for early detection of keratoconus onset and monitoring its progression. And about 14% of the patients with keratoconus do have a family history of keratoconus. The candidate genes which are associated with keratoconus are the VSX1, the MIR184, the dedicator of cytokine 9, superoxide dismutase 1, interleukin 1, as well as the collagen genes such as COL4A3, 4A4, and 5A1. And these are the specific function of the candidate genes which are shown here, which causes the upregulation of the various markers. Now, the genetic conditions which are associated with keratoconus are the various syndromes as shown here. Important in these are the Eppert syndrome, the Down syndrome, and the Ehlers-Danlos syndrome, as well as the Leber's congenital amaurosis, Marfan syndrome, the Noonan syndrome, the oculodentodigital syndrome, as well as osteogenesis imperfecta. There are various inheritance patterns, so that the most common inheritance pattern is that it can be sporadic form or they can be familial keratoconus. And 90% of the pedigrees with the familial keratoconus do display autosomal dominant inheritance. In few percent of cases, other modes of inheritance may be there, including autosomal recessive in families with children of consanguineous parents specifically. There are monozygotic twin studies also, which have shown that there's higher rate of concordance and greater evidence for primary genetic causation rather than environmental etiology. And concordance does support the evidence of heredity as a genetic factor in the etiology of keratoconus. And in cases of discordant for keratoconus reading, eye rubbing and hormonal influences which are suggested as the environmental factors in the etiology of keratoconus. Now, this was a study from Nirayan Netralaya, which looked at the various markers and high level of inflammatory factors such as TNF-alpha, interleukin-6, and MMP9 were detected in cases of keratoconus. There was altered corneal epithelial and stromal expression of specific genes at the corneal cone apex, which drives the focal structural weakness in case of keratoconus. These are the various genomic loci in keratoconus through the linkage studies which have been identified in various populations and various geographical regions. Keratoconus susceptible genes have been reported in different studies which are based on different techniques. And this is just to show the genes which have been identified and the method which has been used to identify them. Overall overview of all investigated genes in keratoconus is this, and again, like some of them are overlapping in the by the various type of the methods used, whereas there are others which are only identified by one or the other method. And genetic screening has revealed latent keratoconus in asymptomatic individual also. So there are 5 variants in VSX1, TGF beta 1 genes, which are identified in 3 Chinese families and 1 Greek family in these patients. It is important to follow the cohort or the families of these patients. And more importantly would be that we do have a multicentric trial, especially in our country or maybe worldwide, because the geographical pattern and the geography might change the various genes which are associated with keratoconus, because there is no causative gene which is known for keratoconus that causes the disease directly. And there's a repertoire of susceptibility genes which has been identified with about 200 polymorphisms in more than 50 genes or loci that confer genetic risk to keratoconus. And available genetic information as of now is insufficient to establish the genetic nature. And there are no specific pathways also which have been confirmed. Coming to the second part, that is keratoglobus again, it is associated with similar syndromes as keratoconus, and possibly this also has a genetic predisposition, although not much work has been done because of the fact that these cases are not too many or not diagnosed too frequently. But a recent study has reported association of variation in TMEM45A with keratoglobus, and especially with progression. Again, pellucid marginal degeneration, the prevalence and the etiology remains unknown, and it affects the inferior peripheral cornea, and combined mRNA expression and patterns have been studied in keratoglobus and pellucid marginal degeneration keratocytes. Again, this is an area where these studies are required, more so because you don't have too many patients. So it will be wiser to pool the patients from different parts of the country or different parts of the world. So thank you very much for your kind attention.
Thank you, Dr. Namrata. That was a very informative presentation. If we don't have questions from anyone in the Zoom meeting, we can proceed on to the next talk. Dr. Tanya from Mexico. She's a clinical geneticist at Hospital Infantil Privado. She's going to be having a recorded presentation on Zoom. Can we put on the presentation now? Audio.
Geneticist from Mexico City, and I feel very honored to be part of the first international conference of Global Eye Genetics Consortium. Today, I am going to talk to you about genetic profile and mutational spectrum of liver congenital hemorrhages in a Mexican cohort. I have no conflict of interest. And first, I will give you a brief introduction about liver congenital hemorrhages, and then I will tell you about the molecular experience of this entity in Mexico. In 1869, Theodor Leber described a blind child with vision loss, wander nystagmus, hemorrhagic pupils, and congenital retinitis pigmentosa. These characteristics were present at or close birth. This German ophthalmologist classified the disease as a new group of pigmentary retinal dystrophy, or tapeto-retinal degeneration. In 1957, a reduced or non-recordable electroretinogram was identified as an essential component in the liver congenital hemorrhages diagnosis. At the same year, Alstrom et al. Reported that in a study from Sweden, 20% of blind children had LCA, which had predominantly an autosomal recessive inheritance. In 1963, Vardenburg et al. Described intrafamilial expression variability and association with keratoconus and cataracts. Liver congenital hemorrhages is the most severe and earliest form of the inherited retinal diseases that causes childhood blindness. This dystrophy is a genetically heterogeneous disease affecting 2 to 3 per 100,000 births, although in consanguineous population or isolated communities may be more frequent. LCA represents almost 5% of all retinal dystrophies and 20% of children with visual impairment in special schools. The clinical manifestations of liver congenital hemorrhages are the following: severe visual impairment from birth or the first few months of life. The patients also present congenital nystagmus, poor pupillary light responses, oculodigital sign, poking, rubbing, and or pressing of the eyes. This sign is also called Franceschetti sign, and an undetectable or severely abnormal full-field electroretinogram. Due to genetic heterogeneity, the fundal features are highly variable as well, but patients usually have symmetrical ocular findings. It may appear normal in the early stage or display a range of abnormalities which can be indistinguishable from other inherited retinal diseases. And these include bone spicule hyperpigmentation, nummular pigmentation, granular appearance, salt and pepper appearance, intraretinal white dots, subretinal flecks, retinal pigment epithelium mottling, or atrophy in the periphery. It is important to know the differential diagnosis. So the different diagnosis of an infant with severe visual loss and nystagmus is broad and includes both syndromic and non-syndromic diseases. So the conditions that are important to be distinguished from LCA include achromatopsia, congenital stationary night blindness, ocular and oculocutaneous albinism, optic nerve hypoplasia, Page's disease, Juber syndrome, peroxisomal diseases, Alstrom syndrome, cobalamin C deficiency. However, a careful examination and detailed investigation, including electrophysiology, usually allow an accurate diagnosis to be established. And molecular genetic testing may then allow a more specific diagnosis. I'm talking about molecular genetics of liver congenital hemorrhages. To date, 17 phenotypes have been described, and mutations in 25 genes have been identified as causing liver congenital hemorrhages. Most are expressed predominantly in the retina or the retinal pigment epithelium. These genes have been shown to encode proteins with a diverse range of retinal functions, including phototransduction, the visual cycle, and photoreceptor development or integrity. As you can see, almost all the genes have autosomal recessive inheritance. In rare cases, LCA is inherited as an autosomal dominant genetic disorder, and mutations in three genes, CRX, IMPDH1, and OTX2, are currently known to be associated with this type of liver congenital hemorrhages. And as you can see, GUCY2D, RPE65, CEP290 are the most frequently mutated genes. And why it is important to have a molecular study? In the first place, to have a definitive diagnosis, since there may be clinical overlap in retinal dystrophies. In addition, it is important to offer genetic counseling, prenatal diagnosis, and for the identification of new genes. It is also important to offer gene therapy to patients who are candidates. And speaking of gene therapy, to date, there are 29 clinical trials for patients with liver congenital hemorrhages. As you can see on the map, most of them are carried out in the USA and Europe and are available for the most frequent genes such as RPE65, GUCY2D, and CEP290. And knowing the importance of genetic study in liver congenital hemorrhages, we decided to do this study called Genetic Profile and Mutational Spectrum of Liver Congenital Hemorrhages in a Mexican cohort. The general objective was to determine the mutational spectrum of liver congenital hemorrhages in Mexican patients. And the specific objectives were identify the genes with the highest frequency of mutation in Mexican patients with LCA, identify the characteristics of the mutations alleles, such as type of variants, location in the gene, and effect on the protein, and identify possible founder effects in geographical areas of the country. On the materials and methods, the patients with clinical and molecular diagnosis of LCA detected from 2005 to 2021 at the Institute of Ophthalmology Conde de Valenciana were included. The clinical diagnosis of LCA was made by a retina specialist based on the patient history of visual symptoms, clinical examination, retinal imaging, and electrophysiology. Clinical and family history details were collected during genetic consultations, and genetic analyses were performed at the Laboratory of Molecular Genetics of the same institute in Mexico City. The methodology used for this study was next-generation sequencing. And as a result, 78 patients from 63 families with clinical and molecular diagnosis of liver congenital hemorrhages were included. 58% were female and 42% were male. 80% were children and 20% were adults. As I told you, next generation sequencing was the methodology used, and in 71 of them, a panel was performed, and in 7 of them, whole exome sequencing was performed. And this is the mutational spectrum of liver congenital hemorrhages in Mexican patients. And you can see that the most frequent gene in Mexican patients was RPE65 with 20.51%. Here in blue, it's represented, followed by CRB1 and RPGRIP1 with 15.38%, then GUCY2D with 12.82%, and then CEP290 with 8.97%, AIPL1 with 7.69%, and then TULP1 with 6.41%, SPATA7 with 3.85%, LR80 and RDH12 with 2.56 each one, and finally CRX, LCA5 and NMNI81 with 1.28% each of them. And 74 pathogenic or likely pathogenic variants were found. Of all these variants, 40.54% were missense, which is similar to described in various series of patients with different ethnic origins. On the other hand, 24.32% were frame shift. 21.62% were nonsense. 9.46% were located at sites of splicing. 2.70% were copy number variants, and 1.35% was a silent mutation. And of the 74 variants, 22, well, thank you for your attention. I am open for new collaborations, so if you have projects in the area of ophthalmic genetics, I will be more than happy to collaborate with you.
Thank you, Dr. Tanya. I think we'll take questions at the end. There is a slight change in order of the presentation. We would like Dr. Divyansh Mishra to go next, followed by 2 talks of Dr. Natarajan. Dr. Divyansh, are you there online?
Yeah, hi. Hi, Somil.
Yeah, hi, Divyansh. So I'm going to give you a brief introduction about you, and then you can begin. Dr. Divyansh is a young and dynamic ophthalmologist, consultant vitreoretina and ocular oncology services at Shankara Eye Hospital, Bangalore. He's also completed his vitreoretina fellowship from there under the mentorship of Dr. Mahesh Shanmugam. He's a very avid researcher, clinician, and academician. He's been a part of many multicentric international trials and co-investigator. as well as principal investigator. He's received many prestigious awards at national and international conferences. He's authored textbooks and had quite a few articles in peer-reviewed international journals. He's going to be presenting to us on melanoma genetics. Over to you, Divyansh. All the best.
Yeah, thank you. Thank you, organizers, and thank you, Karobi madam. Thank you, Natarajan sir, for this opportunity. So, it's our honor and privilege to be a part of this global consortium. So, my talk for today is melanoma genetics. Hope I'm audible and my presentation is visible. So, no financial disclosures. So, uveal melanoma is a common primary eye cancer in adults. Then, usually in a natural history, it usually develops metastasis, and metastases usually have very limited treatment options. The most common melanoma or uveal melanoma, what we see is choroidal, but there are other subtypes like ciliary and even iris. Risk factors, most importantly, are age more than 60 years, gender usually males, and race and ethnicity predominantly in Caucasian, where the percentage is 3 to 7%. Because of the fair skin color and lighter pigmentation, but in India, the incidence is 0.02 to 0.06. Choroidal neva is also one of the risk factors, and oculodermal melanocytosis, like as in this case, we can see the ocular melanocytosis, which we can see in dermal, which is very faintly observed over here. Distance metastasis usually 5 year in 5 year is 25%, and most common presentation is in the liver. And once it occurs in the liver, the survival is very, very less. As you can see, 1 year survival rate is less than 10 to 15%. Other than hepatic is lung, bone, and skin. And what the predictive factors for metastasis, or what are the high risk factors? What we clinically see is large basal diameter, ciliary body involvement, extrascleral involvement, HPE or a histopathology showing epithelioid melanoma or microvascular density having high density or high mitotic rates. PCR-based gene assay, which are more upcoming, still not performed in India. So 3P loss or 1P loss or 6P gain or 8 gain is having a very high risk factor. So COMS has classified melanoma depending upon the size as small as 0 to 3, medium as 3.1 to 8, and large in 8.1 or greater. Clinically, we see that there are different configurations like dome, mushroom, or a collar stud appearance, or a very diffuse kind of an melanoma also.
Divyansh,
Other very uncommon one.
Divyansh, sorry to interrupt. We
Yeah.
can see presenter view. If you can show full slide, it will be much better.
Okay. Sure. Is that okay?
Perfect, perfect. Please
Yeah.
go.
Okay. Sure. So the other is ciliary body melanoma. Again, it is of different types like small, medium, and large, 11 millimeter, 11 to 15, and more than 15. And it can be associated with diffuse melanoma also. So how do we diagnose? Usually the clinical examination, other ancillary tests. The more important ones are the clinical examination, where we see a brownish colored mass. It can have some hemorrhages, and MRI comes to a help, where we can see either a type 2, which is hypo, or type 1, which is hyper kind of an visibility in an MRI. Ultrasound, multiples findings which are important, like high surface reflectivity and medium to low internal reflectivity with a kappa angle. And FFA is not diagnostic, but you may see sometimes a double circulation. OCT in a small melanoma can give us a good understanding, as you can see on the nasal side where the normal choroid is, and on the temporal side where we can see the whole choroidal architecture is obscured and there is some shrem and yellowish colored deposit. And sometimes it can also present into the vitreous cavity. Opta is not diagnostic, but we can study the post-treatment analysis of it. How do we diagnose? By doing FNAB in suspected cases. Either we can go to the transcorneal or a translimbal approach, or either we can go through the pars plana, either by using a needle or by utilizing a 25 or a 27 gauge cutter. And direct transscleral approach is also possible. So this is one of the videos of my mentor doing the surgery. I would skip this for the amount of time. Other option is to do an enucleation if it's a larger melanoma. How do we follow up these patients? Twice yearly physical examination, liver function test, twice yearly chest X-ray to look for any secondaries, and liver either doing an MRI or an ultrasound. Poor prognosis, as we are discussing, older age, larger tumor, pigmented lesion, extraocular extension, and most importantly, the chromosomal genetics, which we would be discussing. So the main genetic feature of uveal melanoma is different from the surface melanoma, as we are discussing over here, that the first mutation occurs in GNA11 or a GNAQ, And then there are other minor mutations which can occur like CYLTR2 or PLCB4. Then there is a second hit. If this hit occurs, then the chance of developing a melanoma is very high. And as you can see, if it's monosomy 3, that is 1A and 1B, the chance of metastasis is much lesser. If it's monosomy 3 with having the BAP1 and SRF2/3 mutation, then the chance of having metastasis or the success or the survival is much lesser. And this is the same slide showing differently. How does the mutation occurs? As we are discussing, the first mutation which is occurring to CYLTR, which causes the activation of this G complex, which is either a GNA11 or a GNAQ, which in turn gets the activated this PLCB4, which causes activation of the further cascade pathway, and where the either the melanosis or either the mitosis of those cells occurs, and there is where the melanoma develops. And how does a metastasis occur? So it is multifactorial. As we can see, it can include either immune interactions like HLA interactions, or either altering signals, as we are seeing that G complex signals, or the tumor hypoxia can even have HIF1 alpha, where it may give either VEGF also. Oxidative stress, again, there are some epigenetic associations along with this metastasis. All that has to be studied. So this is a comprehensive multi-platform analysis which was done in this study by genetic evaluation of metastatic melanoma. In this, the copy number mutation, the whole genome, the INC RNA, the DNA methylation points, and the whole genome sequencing, mRNA, miRNA, and protein. So in this, they were divided into different types of mild to moderate to high chances of having secondaries. So as we are seeing that monosomy 3 with having this BAP1 aberration has a very high risk. And along with that, there are some protein abnormalities also which can be identified. So what are the current established details which are having poor prognosis? As we are seeing, these are all above clinical associations, and below are the one genetic associations, as we are seeing the BAP1 gene mutation, or M3 or 8-1 gain, or 6q loss, or 1p loss. These are very, very important. So there is one more classification, which is 1A and 1B. As we are seeing, the normal melanocytes are usually present. They get converted into the nevus by the first mutation, which is GNAQ1 or GNA11. And then how it goes to the next level, that is a question at this point, which we are solving. But if it goes into 6p1 gain, then it is a little less aggressive tumor, which is 1B. But if there is a chromosome 3 loss, then it goes into class 2A. And if there is a 8p loss or 8p association, then it's a very high risk of having metastasis with epithelioid cytology and high mitotic index of the melanoma. So there is one more classification of ABCD, where again monosomy 3 and then 8p gain or 8 multiple q gains have a very high risk of metastasis. This is one more study showing the same 8q gain, which they have posthumously identified the DNA having association of multiple gains of 8q, which is a very high risk. There are other novel genes which are also right now being identified. Immunohistochemistry also have been studied, but still not completely correlated. So what are the current challenges? Most important, if we have a robust model for prognostication, so that the clinicians and the biologists can give us a correct understanding that how does the patient survive after melanoma, or what is the risk for them, or how do we treat them? So there are many web-based prediction tools, like one by Liverpool Melanoma Prognostic Online, or something from US, which is a model of personalized risk of metastasis in uveal melanoma. So in this is how we need to give the data, and then we need to provide the chromosome status, that what kind of changes are in the genes, and it would give us a risk factor index. There are other newer associations which just by an aqueous tap can be found out, but still those are not cleared by FDA or the European authorities. Again, very relevant, but again, still all those are under evaluation using non-coding RNAs, but still it is under assessment. Roles of infiltrating cells, as we are seeing that inflammation of the tumor and associated inflammation found in the systemic areas are also helpful in identifying the melanoma. So the take-home message from the whole presentation in genetics in uveal melanoma is, first, find better estimator for prognosticating the uveal melanoma, and having association of both the back end and the front end, genomics, proteomics, and proteins, unifying and somehow getting an optimized prognostic model. As you are seeing in European countries and US, they already have it, but in Indian perspective, we have to still develop it by multiple clinical and molecular information. personalized medicine approach, which requires again more genetic and molecular biology association to understand where and how the treatment would be tailored in those cases. So we have just collaborated with one of the companies and we are initiating the study in melanoma, and I would be stopping for the discussion. Thank you again.
Yeah, thank you so much, Divyansh. I think we are taking questions at the end. So
Sure.
do continue keeping logged in and
Sure.
see how it goes. So we go on to the last leg of our presentations from Professor Natarajan. Is he there around? So we are clubbing two topics of his, and not just because there is one speaker presenting both. It's also because both the topics are quite interrelated. One is advantages of 27 gauge vitrectomy, which is a normal vitreous surgery that is done for vitreoretinal diseases. 27 gauge means the gauge has gone still smaller. It's to the tune of 0.5 millimeters. And how you can utilize vitreoretinal surgery as a means of delivering gene therapy for retinal genetic diseases, I guess. So we call upon Dr. Natarajan to present the last two presentations. So, okay, I would like to invite questions while he's loading up the presentation and getting ready to present. Anyone from the audience would like to question Divyansh or any of the previous speakers, as well as from the people who are logged in online?
Hi, Saumil. While we are waiting, let Divyan show the vitrectomy video of the biopsy.
Yeah, Divyansh, if you're still logged in, can you show us that video where you do affinity?
Yeah, sure.
Thanks, Saumil.
Welcome.
So this is a case of a, hope I'm audible and my slides are visible. So this is a case of a suspected melanoma with vitreous hemorrhage. So this is one of the very rare presentations where the melanoma can also present as vitreous hemorrhage. So surgery wise, it's fairly same like what we do as a VR surgeon, remove the vitreous, remove the blood in the vitreous cavity. And most important is the site has to be cleared because there can be sometimes the vitreous attachment above it. As we were seeing that sometimes the melanoma can have neovascularization over the retina or sometimes below it also. And after this, what we are seeing is a cauterization and utilizing the flute needle to go inside the tumor and then sucking the whole the mass of the tumor out. You need not completely remove because in this case, as we are seeing that we are just doing a biopsy to confirm and then only take the further action. So we have an option of availability of brachy with us. And this is what we are seeing is a red laser or a green, not a green, it is a red or a what to say as a TTT, a transpupillary thermotherapy. So in a tumor which is around 3 to 4 mm height, we can use along with that. So once the tumor biopsy has been done, then this was closed with just air because we have made a small opening. Otherwise, the newer gauge, I couldn't edit the most recent one, which sir had done with the 25 gauge, where we need not do this cautery and then make a opening. You can directly jab in either the 25 or 27 gauge cutter into the mass and then activate the cutter so that you would have large chunks of the biopsy. And then rather than aspirating it into the cassette, you can have an external connection of a flute so that you keep the suction connecting in the Luger lock.
Divyansh, that was a nice surgery. Just curious to know what came of the biopsy and then how did you manage it further?
Okay, so in this case, so as you were suspecting melanoma, this case didn't turn out to be melanoma. This was kind of a PHCR, which was, and we had subjected to even immunohistochemistry also for the confirmation. But as you can see, the smaller tumors, once we do TTT, that is one of the treatment options also for the melanoma. So this was a suspected case, but otherwise, in cases which we have a doubt, it does the small biopsy itself can give you an answer whether that is melanoma or not.
Thanks, Divyansh. I think
Sure, thank you.
Professor Natarajan is ready with his presentation. I'll go over to him. So he's going to be first presenting on the importance of using 23 gauge in vitrectomies.
27.
27 gauge.
Yeah, and I'm also going to say thank you, Devans, for this wonderful presentation. And I
Thank you,
remember operating, removing I remember internal resection. So I've done vitrectomy with internal resection and send the patient for further treatment to the oncologist. And I think the patient is surviving. So I'm happy. It took a long time. And as you correctly said, you have to be careful about the vascularity. And I think that was now and then I keep doing, and I also did a masquerade syndrome in 92, which I can never forget. And I know the name of the patient also, Rupali Ingle. It looked like an endophthalmitis. And the child gave me she was a tennis player and I think a swimming champion, almost 8-year-old sub-junior tennis swimming champion. And when the patient was referred and the patient told they had an injury and then came with the endophthalmitis. Somehow I didn't know why I sent a material for culture and sensitivity. What, Saumil? It'll be interesting to know. I'm telling you, culture and sensitivity, and I sent for histopathology. And that was Saturday. And I don't know how Monday morning he received, and Dr. Bishwas did the HPE and called me on emergency. He said, come on, please see whether the child is alive. I said, what happened? He says, you got operated on a retinoblastoma. And I got a shock. Then I remembered back that went through the video. It is I don't know where is it. I was afraid even now my hairs go up. I didn't know what to do because how can you operate on you can't even take a fine needle anymore. Now you are different. But those days in 92, you can't take a fine needle biopsy on a retinoblastoma. I said, I've done a vitrectomy. The patient had something like hypopion. It was actually retinoblastoma seed cell. But then I think I saw you're given credit to Mahesh Shanmugam video. Mahesh Shanmugam enucleated the eye and then did the chemotherapy, radiotherapy. The child is alive and I'm happy. Child is healthy. The other eye is all right. We keep, so then we have done genetic testing also. So I think thanks for your presentation and in continuation, thanks to Alcon for the support. And then I'm talking about this because we use this day in and out. And particularly for gene therapy, we need it, and also for ocular oncology and for genetic studying. So the advanced ultraviolet beveled high-speed probe, and we started with 25 plus, then 23 gauge came. So I'm with them, and I think before Alcon it was Cooper Vision, and then I'm using Cooper Vision from 1984. So I'm celebrating how many years now? 38 years of doing vitreoretinal surgery, thanks to my mentor, Dr. Badrinath. I've also seen VISC-10 made by Golam Payman, operated by Fumitaka Ando and Anatomy, and also my another friend, either 3 generations of them. But they came and did live surgery in Shankaranetara in 86. And they did a beautiful surgery with VISC-10. The problem with VISC is it is a 1.25 millimeter opening. And from that, came to 20 gauge, which O'Malley did, and then Steve Charles popularized it. So I remember doing vitreoretinal surgery, and that time we didn't have fluid air exchange, so we had a 50 cc syringe, and then and Dr. Badrinath used to inject, and sometimes we suck, and the cornea will go down. So from that we have come to ultra modern. You can see 23 gauge. Then we came with a 25 gauge, then 25 plus. And I am happy. I introduced the first 25 gauge in vitreoretinal surgery along with my friend Mark Humayun, who's the pioneer of doing artificial retina. So I always want to bring something new to India. And then we have this valved 23 gauge and 25 plus. And then we have the ultra width high speed 7,500 cut. And now we have advanced ultra width. I think this 10,000 cuts per minute with the beveled tip, 23 gauge and 25/7 in 2017. I'm happy all of them we introduced first. And this is the advanced ultraviolet beveled high-speed probe, is powered by the Alcon's dual pneumatic drive technology. And I think Steve Charles has he's an aeronautical engineer who has and I know only to operate. I'm not an engineer. So, but I learned from him. And I also learned from Dr. Badrinath, you have to have an engineering mind to do vitreoretinal surgery. And from Charles Keepens, I learned you have to be a physicist. He said, you have to know physics. And how do you know otherwise? The entire lens calculation, IOL power calculation, you're a IOL master, everything is calculation, and you have to know physics. And the first Nobel Prize and only a Nobel Prize for ophthalmology is by Albert Einstein. So I think we have to use this engineering technology, and that's why I'm spending time. You see, you have the added versatility, and you can see the different. The tip to the thing is about 0.18 in the earlier one, and then you have the 0.09 in the beveled edge. And it provides increased efficiency. And we as a vitreoretinal surgeon, I know that I have the tactile sensation through the instruments. So we need to have the instrument less than 5 grams, so that you don't have that. Earlier days, we were using 20 gauge, which was heavy. Even the Ocutome guillotine cutter was quite heavy. I think it was 25 grams. So we were operating for 6 hours. I don't know what we were doing. And now we can do it in 15 minutes and 20 minutes. And so we need a improved duty cycle, which provides for more flow for faster vitreous removal versus previous ultraviolet probes. And you have the standard tip with the bevel tip. You can see the angulation and the dimension. You have the 23 gauge and 25 and 27. And I think the problem in India is we reuse it. I know Alcon doesn't like it, but 27 has a fantastic, because you don't look like you operated the patient the next day. And that's the advantage of using 27 gauge. And 27 is the latest advancement in the vitreoretinal surgery. And you can see the various you have the stiffness experience more similar to 25 plus probe than the 25 gauge. And still you have to be careful. Earlier, when we were using the 20 gauge, we used to turn the eye with the instrument. And now you have to make sure you don't turn. You have to exchange the trocar cannula system. So you can see the high flow back flush needle with a diathermy needle. So we need an lens armamentarium to do vitreoretinal surgery. And this is a 27 plus H plus valve entry system. Why I'm showing this is the gene therapy. We need to do all this because now I'm flying on Tuesday night to Belgium to see my friend Dr. Fanny, who's doing robotic surgery to inject Luxturna in the subretinal space. So this cannula removes easily from the trocar after insertion without secondary instrument. You don't have to catch. You can just turn and remove it. And the vent is provided for cases that require a normalizing pressure or a silicone oil or a perfluorocarbon liquid. And here you see a full 27 millimeter shaft length equal to a 25 plus probe for high myopic patients. And you can see you can go far periphery, but again, you should bend, you will touch the lens. So you can exchange the trocar, the location, and you can see the light where you have the 20 gauge to 23 to 25 to 27. And then 27 plus, you have also, the problem is if you have 25, you have to have all this ILM, end gripping, max grip, forceps, everything in 25, and otherwise you have to have in 27. So it increases your cost. That's why I've handed over the hospital to Agarwal. So you have the laser and diathermy accessories, and you have the 27 plus with so many things are required, and I'm happy we have all of them. So thank you very much for this, and I'll go to the next presentation. This one, no? Which one? Which is the third one? Yeah. The next talk is a continuation where we maybe use a 25 or 27 ultra wet probe for vitreous surgery for gene therapy. So what is gene therapy? Introduction of exogenous DNA into a host tissue to produce a therapeutic protein, peptide, or RNA. So I'm very happy to work on inherited retinal degeneration. Many people think why I'm working on this. One is my father, because 84 he started, and my classmate Professor Kumar Manikwel, whom Kelvin Pang said is teacher, who teaches genetics in China and India, and also part of Narayan Netralaya and Adityajyot, and he was originally in Shankar Netralaya. So we can introduce a therapeutic protein, peptide, or RNA. And the history of gene therapy is 1940s, nucleic acids were identified as carriers of genetic information. In 1960s, using bacteriophage, and 1970s, recombinant DNA technology, and 1990, the first gene therapy clinical trial approved for children with severe combined immunodeficiency. In this special issue of human gene therapy, the eye presents a unique opportunity to study the delivery of therapeutic genetic material and why eye is suitable for gene therapy. Arnold Sarsby wrote a textbook in 1950s, which says that ophthalmology is a playground for genetics. More than 200 genes are responsible for inherited retinal disease, and easy access to the site requiring the gene via injection, and partial shield from immune system assures reduced risk of destruction of vector carrying the therapy. Yesterday, we had several syndromes presented, genetic, which says 90% of the genetic disease has some involvement because development of eye, we have the neuroectoderm and surface ectoderm, which merges and produces the anterior and posterior segment. And so the development is so good, so unique, and every structure of the eye is different and every pathology is different. So it's very interesting organ. And second, we all see because of the macula, the center of the retina, and large number of retinal degenerative disorders are recognized principally affecting the photoreceptors. And because of that, we see the acuity of vision, the bright vision, and the color vision, and our retinal pigment epithelium in the outer retina. And when I introduced OCT first in India, we had this diagram like this, photoreceptor, RP, and 10 layers of retina. They thought, what is Dr. Natarajan doing? Even Dr. Badrinath asked me, what are you going to do with this another playing instrument? I said, one day something we'll do. And now you can see even a general practitioner, general ophthalmologist wants to buy an OCT to study the layer of retina. So the common inherited retinal dystrophy, which comprises of millions of patients in India, diffuse photoreceptor dystrophies, RP. RP itself, I think 500,000 patients are there. Leber's congenital amaurosis, cone dystrophies, totally misdiagnosed or wrongly diagnosed or not diagnosed at all. And macular dystrophy, Stargardt dystrophy, Vitiligo dystrophy, familial Drusen pattern dystrophy. Stargardt was a professor and chairman in the University of Marburg, and I'm happy I'm also along with him in the Retina Hall of Fame. In both types of disease, vision loss occurs primarily because of progressive retinal photoreceptor death. The need for new treatment strategy is compelling. And gene therapy, identification of new target genes, development of viral vectors that permit delivery of large gene payloads, initiate strategies to treat conditions that have polygenic basis, to develop programs that not only rescue patients with the existing condition, but therapies that delay, prevent, or permanently cure a disease. There's no cure. Thanks to Subur Huang, who is my friend. I think there's a big function going on. He's a part of the Retina Foundation, which gives award for people who work on preventing blindness. And how retinal gene therapy works, the vertebrate neural retina contains many different types of cells like the retinal ganglion cell, rod and cone photoreceptors. In retinal gene therapy, adeno-associated virus called the AAV enters these various types of cells and expresses the DNA sequence of intended therapeutic value. The few approaches are the current state of the art in gene therapy involves the delivery of various types of viral vectors into the subretinal space. And that's why you need surgeons like me and Saumil to effectively transduce diseased photoreceptors and retinal pigment epithelium. Nobody thought about it. And I remember Ambati called me one day and said, do you have any scientists who want to work only on retinal pigment epithelium? I said, how do I find in India? And I couldn't. And now I found that the guy from Shroff Charity went to the Ambati and worked, and now he's working in Delhi. I met him last week in SARC meeting. So subretinal injection is becoming, it's not just an injection. You have to do a complicated vitreoretinal surgery with the armamentarium instruments, which I showed. And that's why I gave that talk, not only for my financial interest of collecting funding for this conference. Subretinal injection through vitreoretinal surgery is becoming increasingly popular among researchers and clinicians. To date, several approaches for doing this vitreoretinal surgery and delivering the gene and the vector in the subretinal space have been described in scientific literature, all of them effective in accessing the subretinal space. And the most common methods to deliver, which it didn't work, is I'm happy it didn't work, because otherwise general ophthalmologists would have taken over, like Dr. Meenu, which I don't want. And it has to be, we have to be unique, and we have been trained ourselves to be unique. What, Saumil? So that we should have that access to the retinal pigment epithelium. And then a delivery of, so I'm going to change it, Dr. Afros. Next time you change this to vitreoretinal surgery to deliver subretinal targeting the photoreceptor and IRP cells, and also access the suprachoroidal space. And which is a gene or cell therapy protocols that deliver viral vectors into the subretinal space typically involve a vitrectomy. That's why I'm talking this, followed by the introduction of a therapeutic agent. And doing vitreoretinal surgery is not simple because the patients may not have a thing called posterior vitreous detachment. That means the vitreous is attached, and particularly I remember operating a retinitis pigmentosa patient with retinal detachment, and then it was there was absolutely no PVD. So to induce PVD is another research we have to do into the, and then inject this therapeutic agent into the subretinal space via the smallest possible entry. You saw Dr. Devanshu mentioned about 27 gauge, and we are actually entering it through 33 gauge. Nowadays, I give intravitreal injection using 33 gauge needles from US. Amazing. Patient doesn't feel at all. The subretinal injection via vitrectomy is carried out under retrobulbar or GA. In India, we do it under retrobulbar. And sometimes I've tried under topical, but it's terrible for the patient. So better to do under the retrobulbar or the peribulbar anesthesia. And 3-port pars plana vitrectomy done using the 23, 25, or 27. And after performing core vitrectomy, posterior vitreous detachment, which looks simple but is not simple, that can produce complications of vitreous tear and then retinal detachment. And with you have to use triamcinolone for visualization of the posterior hyaloid. And this is how 3-port vitrectomy, because most of you are scientists and maybe you have access to doing vitreous surgery in some wet lab. But that's an advantage. Nowadays, you can try in the wet lab, in the slaughterhouse pig eyes. I think anybody can try. But you can definitely, all researchers working on gene therapy should do that, whether you're a MSc or a biotech. In a pig's eye, from a slaughterhouse, you should do, not in the live animal. And you can use this to see how a vitreoretinal surgeon has to use this 2 eyes, 2 legs, 2 hands, and the brain too, and the ears too. No? You have to hear the sound. And after that, peripheral vitrectomy is completed. The superior vascular arcade, which avoids blood vessel, is the optimum location for gene therapy. Preoperative planning using OCT, autofluorescence have been taken into account in order to avoid areas of retinal atrophy where the retina is less susceptible to detach. And you see, this is the area, superior temporal arcade, areas of retinal pigment epithelium atrophy, areas indicated for the delivery of the subretinal gene therapy. And then you see fovea is also marked, and this is from the reference given below. So you do the delivery either using a 38 or a 41 gauge. You can see that's smaller than the size of the hair. And the appearance of pseudoschisis, which is described as momentary hydration of the outer plexiform layers on OCT, can be brought on by beveling the subretinal cannula. Therefore, the surgeon has discretion over whether to bevel the cannula or not. And we can also use the nowadays the intraoperative OCT for this. The non-valved trocars are recommended, which I showed in the previous presentation, and the technique can be performed in 1 or 2 steps. And the 2-step variant used by McLaren, which I hope the video is there. And this way, the creation of space for the, and that's where from the manual Dr. Robert McLaren did, now they want to do robotic to do that in the subretinal space. Volume. This is from Robert.
For sufferers of hereditary blindness, there may be a light at the end of the tunnel. 2 experimental trials have shown that gene therapy could one day cure a rare form of blindness that affects about 2,000 Americans.
41.
The research teams tested the treatment on a total of 6 patients. Here you can see how they performed the eye surgery. Repair genes were injected into the eye of this young visually impaired patient. The eye was infused with air, then reinfused with fluid near the end of the surgery. To measure the patient's performance before and after the treatment, he was guided through an obstacle course. Here you can see how he navigated through the maze prior to the surgery. He bumped into obstacles 6 times and was disoriented twice. It took him 77 seconds to finish the course. 6 months after the treatment, the patient showed a dramatic improvement. He was able to make his way through the maze in 14 seconds without bumping into anything or getting disoriented. He was one of 4 patients that showed an improvement. The treatment also proved to be safe, which has not been the case with previous gene therapy trials.
So as you see here, one you see Dr. Jovanovic is the second mentor, and he told the video surgery is not developed because of the space is dark and it's difficult to access. And I'm glad I introduced the wide angle observation system to most of the world, including India, in 1990. It was invented by Mark Spitzness from Bonn. And unfortunately, I don't know why it was not popular in Germany too. His student Frank Koch then later made it. He's now professor in, and I'm a visiting professor there in Frankfurt, where the wide angle observation system, and now we have ultra modern, almost fifth generation has come, and we have used all that. So the idea is to inject, or it's a deliver subretinal of 0.3 ml being delivered into the subretinal space for RPE65 related inherited retinal degeneration, and 0.1 ml in cases of choroideremia. And you have to measure that. So this is there is a controversy regarding foveal detachment with subretinal injection due to the unforeseeable behavior of the retinal tissue in inherited retinal degeneration. And manipulation of the vitreous without vitrectomy is often complicated by the development of retinal detachment, and therefore most procedures involve access to the post segment, including delivery of the material into subretinal space, are performed in conjunction with the vitrectomy only by a vitreoretinal surgeon like us. The reason is you have to avoid complication. Surgery means complications can happen. So the most experienced surgeon will have the least complication. So what's new? Recently, new research have been developed for subretinal delivery. Wood et al. Describes access to the subretinal space without vitrectomy with a nanovitreal subretinal gateway device designed by Vortex Surgical for the management of the SMH, and this device could also be used for the subretinal delivery of the gene therapies, avoiding the risk associated with general anesthesia and vitrectomy, especially in pediatric patients. And these are the references. Thanks to again Dr. Jaydeep, Afroz, and Samir for making this presentation. Is there any other presentation I have to do, or any questions?
That was an excellent presentation, sir. I think we had said that we would take up questions at the end.
Okay.
So for all the talks that we've had so far in this session, if anyone from the audience or from the Zoom live has any questions to ask, this would be a good time.
What is your comments?
So that was an excellent surgery, sir. My comments are people dreaded the most difficult disease in the eye to treat was known to be retinal detachment. Now people are actually inducing retinal detachment to deliver gene therapy. So it's no longer considered to be dangerous. And we have far better instrumentation to actually tackle it.
No, but I'm telling publicly, we cannot make it a simple procedure just because it's an experienced scientist. Sir?
Is the technique done in India?
No, government of India is not approved. We can't get the gene, the vector, because only one treatment is available, Luxturna, and Vortagen, the name where we need approval, which we are not.
I think it's a 5 crore treatment, right?
8 crore now.
8 crore.
$800,000.
So and the result is unknown. It's not.
Dr. Meenu is telling she's got funding.
Okay.
Maybe sometime next year, we are planning to start a gene therapy center at Somaya. But I don't know how far successful we are going to be, but we are trying and hoping for the best.
Before that, I'll start.
Yes, definitely.
You all can have a tie-up, definitely.
No,
that
sir,
I told
get
them,
it, sir.
but we are talking to, I know the whole 3 generations of, what is the name, Somaya, but they want money. And I'm not interested. But I will take money. No, they have to pay me to come to Somaya.
Yes, sir, definitely. No? Yeah.
Any other point? No, I think the main thing is I want to create interest. I hope the patients are also watching. Probably tomorrow they will be seeing from the YouTube. So whole idea is to say that therapies are available in the world and India is still behind. But one day we'll probably, we are like the turtle and the rabbit, and one day we'll win with more newer therapies, indigenous way, and our own way. I think we should do. I think we have a lot of scientists working, and I remember somebody working on photoreceptor from IIT Kharagpur, and recently I think there's an Indo-American grant between IIT Kanpur and US on a photoreceptor something, and I think how many $1 million, I don't know. So no, they're working, but the problem is we are not having access. Like this, Dr. Meenu madam has to come to me and or you and tell, but she's keeping it secret. It's a problem of India. Everybody wants to. That's why Alexander the Great came and conquered India. British also came, divide and rule. No, we are not united. I hope the United States of America, we learn to be unite. We are not united.
So
And I'm open about it. I'm not telling only you. I'm not telling only about Somaya. Every institution, every institution say my institute. No, I don't want to do joint work. And if you see, Jay Chaplani is doing a great job of asking several people. I'm telling the same. I'm requesting Saumil. Saumil doesn't have time for me. And I hope Shruti is there, Samruddhi. They all out. I'm developing them. I said they should do all this for first in the lab, our own lab. Next session, who's chairing? What is the time? Next session is at 4, no?
Session is at 4 o'clock.
Already it's 4 ho gaya.
Yes, and Dr. Kirit Modi is the chairman.
But sir,
And convener is Dr. Chaya Shinde. I think she is already.
Chaya Shinde, madam, you can take
and
the chair.
Mayur as well. Mayur,
Mayur, I saw Mayur also. Chaya, madam, thank you very much for coming. And Hitesh also is there.
Yeah,
So
Hitesh
I
has
think
been-
Anuprita was here. Come, please come. Now, Kirit Modi, sir, I don't know, somebody has to call. Then Hitesh is there, Chaya and Mayur. Joseph, you're calling Dr. Kirit Modi? Okay. Except me, I'm inviting everybody to start the session. To whom? To Saumil? Saumil, Suprabha wants to tell something for you. Now you have to be there only. She'll tell in the mic only.
Requesting sir to give moment to Dr. Somil sir. Yes, sir.
It should be vice versa. So you've got so many moments.
For you. Thank you. Thank you. Thank you.